Welcome to Atoms to X-rays, a series about new developments in the physical sciences. Atoms to X-rays is brought to you by the Division of Physical Sciences and the San Diego Foundation's Reuben H. Fleet Foundation Fund, sponsors of the Atoms to X-ray website for teachers and students. Please visit the site and let us know what you think. We hope you enjoy the program.
To introduce our speaker, Mike Sailor, some of you have probably seen him in the news and newspapers and magazines since September 11th from the work that he and Bill Trogler and their colleagues in their labs did on developing new materials, or new sensors for TNT and nerve gas. And he is here to talk about sensing chemical and biological compounds using nanomaterials. Mike?
Thanks, Kim, and thanks for inviting me to come speak to you today. What I thought I'd do is I want to start by setting up the problem. A lot of people are familiar with the concept of sensors. Many people think of a sensor that might be in your wall. Your thermostat has a thermometer in it. That's a sensor that determines whether or not your air conditioning or your heater needs to go on or off. There are many sensors in cars. For instance, there's a sensor called an accelerometer that measures when your car crashes and it ignites your airbags when the car stops suddenly. The sensing that I'm going to be talking about today is primarily sensing of chemicals and biological molecules. So it's not trying to look at temperature or pressure or anything like that. It's all really chemistry. And the slide I wanted to set this up with, to start with, is this picture of taken out of The Economist a couple of years ago. In recent months, we've been very concerned with chemical and biological warfare agents. This is not a chemical or a biological warfare attack. It's actually an industrial accident, and it's not a biological agent that was used as a chemical agent. It turns out that what you're looking at here is a bunch of dead fish, and they were in the Danube until they died, and they suffered cyanide poisoning. And it turns out that upstream, somewhere in the former Yugoslav Republic, there were some mines where they were using cyanide to leach precious metals out of the rock, and a dam broke somewhere, and the cyanide spilled out into the waterway and caused all the fish to die. Of course, if it's really a chemical or biological attack, it may be something that attacks agriculture or livestock. It may also be something, and as we're very aware of nowadays, that may be attacking people. And so there's a lot of need in the field, in our homes and in our doctor's offices, to have sensors, things that can detect whether something is toxic for an animal or a human, whether you've eaten something that's toxic, whether you have something, a certain disease condition that needs to be treated. And so that's kind of the area that we play in. And sensors is a very familiar area to most of the medical community. They use them all the time, and typically what they use are medical sensors or biomedical sensors. And they're usually based on two different types of molecules, and I've shown examples of them here on this slide. And it really serves to illustrate the first part of the problem. What I want to do is split this problem of sensors up into two areas. The first one is recognition. So how do you know that you've seen something? And in a doctor's office, if you're given a test, they might use something such as an antibody to test whether you have a certain disease, because that antibody would be able to bind very specifically to what's referred to as the antigen, or the thing that the antibody targets. In your body, when you get sick, the antibodies move around and find the foreign molecules or chemicals, and those antibodies then attach to them and then trigger the body's immune response. Another form of recognition used by molecules and used by nature is how DNA is put together. You've all heard or familiar with the familiar term, the double helix. The double helix refers to DNA, which is the programming code for all of life. It comes in two parts, two strands, and those strands come together to make a double molecule. That's the double part of it. and it forms a helix, and that's the helix part of it. Those two parts are very precisely engineered by nature to recognize each other and only each other and nothing else. And so nature has evolved over the space of about 2 billion years techniques for molecules to recognize each other. And one of our challenges, and the challenges that are taken on by the medical community and used and harnessed every day, is to take some of these chemicals, some of these molecules that nature has provided to us, and use them for purposes that nature didn't intend. Typically, we're using them to provide us with some indication of whether we're sick or healthy. And of course, these things usually are embodied in very large boxes. This is an example from PerkinElmer Applied Biosystems, a box that is designed to analyze DNA. So that's the second part of it. There's a recognition aspect, and then, of course, there's encapsulating it into some sort of a widget or a device, and we'll talk to that in a second. But before I get to that, I want to talk about the second part of recognition, which is something that's also used very commonly in nature, and it's really the second class of recognition. If you don't want to go specifically looking for some target molecule, some very specific other molecule, then what nature does is it defines other systems that allow you to do recognition of broad classes or building systems that are able to be trained. And the one that we're most familiar with is the olfactory system, or the nose. Your taste buds, your nose have both in your tongue and your nose, you have the capability of determining certain types of molecules. And the really neat thing about how the dog's nose or the human nose works is that it doesn't really have a specific receptor up there for perfume and Coca-Cola and toxic agents, poisons such as spoiled food. What you have up there is a bunch of receptors. Turns out there's about 1,000 different types, and somehow your brain is able to train itself for a specific response or a given molecule or something that you might breathe in, goes into your nose, and it triggers responses in all these 1,000 different types of sensors in the olfactory bulb, and sends some sort of pattern to your brain, and your brain eventually is trained and recognizes that particular pattern. And that's kind of good. For instance, dogs never had any interest in detecting explosives out in the wild. And there's no reason that a dog or a wolf or its ancestors needed to smell TNT. But we can train dogs to smell TNT because they have the capability physicochemically inside that nose. There is some receptor, some little widget that will respond to that molecule and will trigger something that goes on in the brain in a variety of different patterns, and that pattern then gets recognized and trained into the animal, and that's how they can be trained then to smell or go detect things like explosives or drugs, anything else that you might think that you really don't, that there's no genetic reason, predisposition for these animals to be able to detect. And it turns out that this process also has been implemented very well artificially by us, researchers and scientists, and I'll talk a little bit about that in a few minutes. And one of the last issues I want to touch on before I get into some of my talk is there are a number of problems in taking something from a laboratory. Let's say we develop our new kind of taste bud or new kind of recognition element for anthrax. We've got to then bring that thing into some package, and that's primarily engineering, but it starts out really at the top of the list as sort of the science. How do I build my transducer, what we're going to be calling a transducer, the thing that does the recognizing, the taste bud, the individual sensor element. How are we going to collect that? Your nose collects it by breathing in. It turns out that's a real challenge right now in terms of trying to detect small numbers of biological hazards in air to be able to collect a lot of sample and present it, concentrate it down and present it to the sensor. And again, this then feeds right in heavily tied to engineering, how we integrate these kinds of systems into devices and into an infrastructure that would be able to respond. Either in a doctor's office, it's a doctor reading a readout on a machine that tells him or her that there's a certain condition or certain concentration of some antibody or antigen in the human patient that they're trying to, then they decide that they need to treat that. We don't really have models or protocols really yet in place to detect things or to respond very efficiently or effectively to biological or chemical hazards that may be coming from, say, for instance, terrorist activities. But we're pretty good at responding to hazardous materials, spills, and whatnot, and that's something I think that over the next couple of years will have to be developed and grow. And the last thing that's really kind of not too relevant to my talk, but I did want to bring it up, is that there are big financial issues involved in developing sensors. For the medical community, there's no problem because everybody wants to live longer. And so there's lots of money out there to develop new kinds of sensors that might be able to detect if a patient has AIDS, or very quickly in very early stages detect if somebody has cancer. There aren't quite the size of the markets for doing monitoring of chemicals, such as that cyanide spill that I mentioned earlier. If they were able to detect that cyanide in the water earlier on, maybe they could have remediated it, maybe they could have found the source quicker. The model there, there wasn't really a financial benefit to have cyanide sensors or people taking samples in the Danube, constantly. And so how do we do that? How do we deal with that? I think also that's going to be dealt with sort of as illustrated in these little pictures down here, taking some science out of the laboratory, putting it into a box and trying to see if it works out in the field, and then ultimately miniaturizing it to something that's the size of something you can put in your hand, and as I'll get to at the end of my talk, maybe something that you can't even see, it's so small. And what we're going to be relying on in all of this, I think in the next several years, is piggybacking onto the semiconductor technology industry. And they've developed these wonderful techniques to build very small, very lightweight, very low power devices. We call them computers. And our challenge really is to try to take some of the systems that can be put onto a chip. In the case of a computer, it's transistors, the things that do the thinking, and convert them into things that can be doing our sensing. And there's a lot of examples of that already out there. And I've got a couple of pictures from two different companies that kind of represent that. Again, this is just kind of restating what I've been saying. We're going to break this problem up into two parts. First, there's our recognition element, and it may have some biological recognition capability, which is sort of drawn by these greenish-brown blobs. It may not be, and I'll show you some examples of how we can do very simple things to do sensing that don't involve using biology at all. But that's the recognition part. And then transduction is, okay, now we're going to have something happens. These two molecules find each other. How do they tell the outside world? If the dog smells something that he likes, he wags his tail. So we want to have something that allows us to see this dog's tail wagging that is triggered by, ultimately, can be tracked all the way back to this fundamental chemical recognition event. And that's the transduction side of it. And what I'm showing here is an example of how transduction has been done, for this example, in DNA analysis, where the group at Nanogen has developed on-chip devices that instead of measuring a current through a transistor and doing computing, they measure a current, or they run a current through a chip and do a DNA analysis. And the little pads, instead of being transistors, have DNA molecules parked on top of them. And when recognition occurs, they get a signal and they measure it. Now I want to spend a couple minutes talking about how really the differences in these problems from the chemical and the biological side of things. People have been very worried about chemical and biological warfare, and often it gets lumped into the same concept. The anthrax scare that everybody's very familiar with wasn't a scare, it actually happened, an anthrax event. Is that the same as cyanide getting dumped in water? Well, it's a terrorist attack. It could be an accident. But how we approach detecting that and determining whether we've seen that event is a very different problem. And it really boils down to this. Molecules are very small. The molecule I'm showing here on the left-hand side of the screen is sarin. It has a size of about 1 nanometer. And anthrax, the spores that were in those envelopes, have sizes much larger, on the order of about 1,000 nanometers. So about 1,000 to 1 difference in size. And so that's the kind of analogous to the difference between a pea and an elephant. And what we'll get to in a minute is that molecules are much easier to detect. They're much harder to protect against. And this can be shown on this example here. If there's a cloud of sarin in this room right now, we would have to have rubber suits on. Sarin as a molecule will pass through your skin. It also, of course, will get in, you can breathe it in, and that's where you're really concerned. to avoid getting that sarin into your lungs requires full face shields, gas masks, cartridges that can filter out, not just filter out the particles, but actually chemically react or absorb these molecules in the cartridge so it doesn't get into your lungs. And so the molecules are small, they get around things, and they're very hard to keep from getting into your lungs. Whereas small particles, say as anthrax, it turns out that they're much easier to protect against. You could use just a surgical mask. In fact, that's why often surgeons wear masks in the operating theater. Is either they might have some disease that they don't want to transmit to the patients or vice versa. And these are fairly effective in doing that, and that's because a lot of the larger things like bacteria, and in the case of anthrax, the Bacillus anthracis, is a fairly large particle relative to molecules, and it's fairly easy to protect against it. It turns out that although it's easier to protect yourself against something like anthrax, it's much harder to detect it. And it's sort of the dichotomy in this game. And the chemists have developed tools to detect molecules, and we'll talk about some of them in a few minutes, based on what we call spectroscopy, taking different wavelengths of light or different types of energy and passing them through these molecules. Different kinds of chemical reactions or catalytic reactions that can be developed to detect molecules. And this isn't just sarin, this is just an example. It's true for things like cyanide, botulinum toxin, a number of things that we would sort of class as molecules as opposed to organisms. Whereas if it's a larger thing like anthrax, turns out anthrax spores to most detectors look very much like pollen. And to really be able to determine whether you have that little blob there is anthrax requires the tools that I've already mentioned, using sort of antibody-based or DNA-based analyses. And these tend to be slow. They tend to take 20 minutes at the best maybe to crack a spore open, dig out its DNA, and figure out that it's actually anthrax and not pollen or a soil bacteria or something. And so that's the challenge there for the biological detection, I think. So what I wanted to do now is just back up into these two parts, and we're going to start talking again now about the transducer side of things. And I thought what I would do is do a live demonstration of a sensor. I brought one here. You can buy these at Radio Shack. This, or any other electronics store really, this is a voltmeter. It has a mode that can be turned to that measures resistance. And I've done that. And somebody want to help me with this? Anybody got Kim? Well, since you introduced me, Kim. All right, so what we're going to do to Kim here is, let's see, come on up here. All right, I want you to, these are the two leads going into this voltmeter. This thing will measure resistance. And so in fact, we can touch it to a piece of metal and it'll make a measurement that's very low. And we're going to touch it to Kim and we're going to measure his body's resistance. Okay, now, what do you read there, Kim?
0.70, 0.7.
Okay,
0.6.
now squeeze it harder.
Wow.
Went down to 0.4.
0.4.
Right. Okay, so what we're measuring here with this is just the resistance of, turns out, Kim's skin. If I didn't want to keep you here long enough to ask you this question, Kim, but it turns out that you can, people have heard of the polygraph test. So polygraph, poly means there's a couple of different things they're trying to look for there. One of the measurements they make in a lie detector test or the polygraph test is what we just did, the resistance of your skin. And so this is a type of sensor that can detect, in the best case, whether somebody's lying or not. There's a lot of argument about whether that would really tell if Kim is lying or not, but that's a simple transducer then that we're using. That is, in this case, little pieces of metal and the voltmeter to determine whether or not Kim is made out of flesh and blood. It turns out that this is actually on our website, so this is something you can look up and build for yourself if you want. You can modify some things a little bit and make things that'll sense things more than just skin resistance. This is a capacitor. You can buy these things for a couple of pennies, again, at an electronics store at Radio Shack. So a capacitor has, I'll describe its function in a little bit, but it's basically two plates and some current can pile up, electrons can pile up on one plate, and it's used in electronic circuits. take a capacitor, cut the top off of it, and this is what you would get. And if you look inside this thing, capacitors have two plates that are parallel. They're metal conductors. And what I've done is just clip the top off of this. So these two metal plates are exposed, and that's what we have here. With these little metal wires exposed, if any water gets adsorbed from the air onto those plates, if the plates are close enough together, then electrical current will pass. For the same reason that you can use electricity to electrolyze or split water, current will flow through this water. And so you can, in the best case, when the things work, you can actually measure the relative humidity of the room or the outside air using a device as simple as this. And this is, as I mentioned, it's on our web page, and you can make it a little more sensitive by adding a transistor and a couple of other little circuit elements into this. That's the little capacitor structure right there up in the upper left. And this device will work most of the time, and you can use it to make a measurement of the relative humidity of a room. You can also use it, you can stick it in your planter box and decide when you need to rewater your plants. So that's a simple example of a transducer, and you might think this is kind of trivial, but in fact there are companies that make devices that are based on this concept. I've got a picture of one of them here. This is a company called Cyrano Sciences. Cyrano comes from the fact that they mostly try to detect things in air, and so it's kind of the electronic nose. And the technique that they use is somewhat similar to what we just described. They have two metal contacts, and they run some current through some stuff. And the stuff here has carbon in it that conducts some of the electricity, and there's a polymer block here too. And they measure resistance through these little buttons, and when they're turned on, current flows. And then when they expose it to some kind of a gas, the wires break and the current drops off. And it's shown in this bottom trace here. So what's going on there? Well, the reason I showed this is because I want to illustrate my next concept, which is the concept of recognition. So we talked about transducer. We make a measurement of conductivity. In this case, if we want to make this thing specific to something besides water, we got to play some games. And the game that was played by the Cyrano group was to encapsulate their little conductors in a polymer matrix. This is a polymer or plastic material. You may have found one day, if you were playing with your saran wrap, when you put it in the microwave oven, it gets soft and melts. Sometimes if you put polymers into organic solvents, like perfume bottles have to be made out of certain materials, because if you put the perfume or the rubbing alcohol or a number of different kinds of liquids, chemicals, in the wrong plastic, they'll melt the plastic. And that's kind of what they're doing here. In fact, they don't melt the plastic, but the plastic that they use will swell when it sees certain kinds of gases. And they change the plastic to have the plastic swell at different rates and to different amounts. And so this is a system that could measure hydrocarbons. And the reason it can measure hydrocarbons is because these polymers are very greasy, and when they place the polymers on the surface of this sensor, they have an affinity for other kind of greasy molecules. And this is basically the concept I want to illustrate that they're using here, they're harnessing, is a concept we use in freshman chemistry. You may have heard of like dissolves like. And this is an example that I give in my freshman chemistry lectures sometimes of ethanol dissolving in water. And this is an ethanol molecule here. And there are chemical bonds or different kinds of molecular interactions that occur between the ethanol molecule and water. In this case, I'm illustrating the hydrogen bonding that occurs in the water-ethanol mixture that causes the ethanol molecule to have some affinity for water. So you know that you can buy alcohol in water. It's like beer or vodka. They have different concentrations of alcohol in water. You can mix alcohol and water to any amount. They're fully miscible, that's what we refer to as being miscible, meaning that they can make a single mixture just by adding different amounts. They completely mix up with each other. And that's very different from, say, something like oil on water. You're familiar with putting oil on top of a bottle of water. The oil will actually sit on top of the water. It won't dissolve in it. And that's an indication of the fact that the water and the oil do not like to mix with each other, and the interactions there are not favorable, and so they separate. And that's the same concept that's being used in this little sensor here. The polymers will pick up certain molecules, and other molecules they'll reject. Some of them they mix with, and some they won't. If they mix with the polymer, it turns out that the polymer swells a little bit and these little wires break, and they can measure changes in conductivity and make a sensor out of that. Now, I wanted to then talk about another concept that we've been using in our lab in sensing to take advantage of other kinds of interactions of molecules with surfaces. And I want to illustrate that with this slide here in this movie. What you're looking at here in this film is a small piece of a silicon wafer that has an optical structure built into it. And you'll see what we're doing in this experiment, or in this little movie, is it's sitting in a glass chamber, and we're opening a valve and letting some gas in and out. And as the vapor comes in, the color of this little film changes. And as we pump it out, or we open a vacuum tube to it, and we suck the vapor out, the color of the film will change again. And in a second, you'll see it goes from red to green. And what's happening here is an optical transduction mode rather than an electrical transduction mode. So now we've been talking about using electronics up to this point to do sensing. This is a way of doing sensing with optics. And it also takes advantage of using a material that we refer to as porous silicon. This is a film of silicon on top of a silicon wafer, just like in a computer chip. But these films, turns out, they will condense molecules inside their pores. If the molecule, in the latter case, I was showing an example of how a polymer or a gas can diffuse into a polymer. But these materials actually have porous structures, little films, that will condense molecules. So molecules just diffuse right into the holes and just stick there. And that phenomenon we refer to as capillary condensation. So it's a little bit different than the process of molecules going into polymers. And there's a lot of heavy lifting intellectually we do to try to get these sensors to work properly. And this is primarily the thesis work of Ting Gao in my group. But what she figured out was that we can tune the holes of these little films that we make. These films have very small holes, so inside these holes, molecules can condense, can get in. And it's very similar to what happens with the small capillary. If you take a capillary of glass and you put it in water, the water or a straw, the water will wick up the straw a certain distance. This same process happens in the nanoscale. When it happens, when we make little straws that are only a couple of nanometers in diameter, what happens is instead of just water rising up a hill, water in gas phase will actually turn into liquid. You can cheat the system. You can cause molecules that think they want to be in the gas phase to condense. And it's a phenomenon referred to as capillary or microcapillary condensation. And that was the process that's occurring that gives rise to that color change in this movie that I showed here. The movie, again, shows these films being exposed to, in this case, an acetone vapor. The acetone gets into these little holes, the color of the film changes. And the reason the film color changes is a very similar process to how oil films on water will have certain colors. When molecules get into these holes, they change what we refer to as the refractive index of the film, and that causes the color to change. Very much like how when the wind blows, you have a little puddle of oil on top of water, the color of the oil may be purple or green or red, and as the oil film changes thickness, its color changes. It's the same kind of phenomenon. And it takes advantage of the fact that molecules like to be in small places, and so they will condense and turn into liquids. And that's the nanotechnology part of it. We can take advantage of making small devices by using semiconductor technologies, and I showed you in this last example of something that can be integrated onto a silicon chip that would do sensing instead of electronics for you. And just like in electronics, there's this big driving force to making things really small so that, ultimately you can have a lot of processing power in something the size of a cell phone. Everybody sitting in this room who has a cell phone also has a sensor on your belt. What the sensor is telling you usually is signal strength. You look into your cell phone, usually there's a little bar graph there that tells you how close you are from the transmitter. That's a kind of an electronic sensor also. And the great thing about that sensor is that you just kind of get it along with the rest of the box. But a couple of years ago, to get a box like this that would talk to the outside world, the box would have had to have been this big. And years before that, we didn't even have them. And this driving force to making things smaller and smaller is what's really important for the sensors area, because ultimately if you get something that's small and cheap and runs on batteries, you can put it out in the field and have it smelling for cyanide in that water supply without having to be out there yourself taking a sample. And that's one of the big motivations for the sensors area right now. And so that's great if you make something the size of a cell phone, but how much smaller can we go? Can we make something that's the size of a particle of dust and tell it to do something for us? And that's what the last part of my talk is going to be about. And what I'm showing you here are some of these same little optical films that we've been making in our lab, but these are made patterned on a wafer by Yang Li to make some very small, little responsive particles. We call them smart dust. This little square that you're looking at here is 80 microns, 94 microns actually in width. So that's about as big as one of your hairs is round. We carve these things up into little plates, and that shows a picture of one of these freestanding plates. We make these things into little particles, and these little colors will change when they see gases, as I showed you earlier. But we can take the detector side out of it, and we can make these things into particles that are so small that you can barely see them, and they're little. That's why we call them dust particles. I have a little beaker of them here. It's the same beaker that's over there. You can come up and take a look at it a little bit later if you want. They're really beautiful. If you get a good shot of them, they just glisten in the sunlight. And so what are we going to do with that? Well, that's our sensor. Our transducer is that particle, and our detector for this, or the transducer, the other half of the transduction, is going to be done from a distance. And the reason we wanted to do this was we wanted to be able to develop techniques that would allow us to make sensors that are very small, lightweight, zero to low power, and be able to probe them remotely so that you don't have to be sitting on the side of the building when the sarin gas flies by, or you don't have to go out into that estuary and be constantly taking data. You can maybe have something on a telephone pole shooting a laser beam down and monitoring it and seeing whether that little film is changing color. And that's what we did in this experiment. And it also illustrates this hydrophobic, hydrophilic concept that I brought up earlier, and so I want to talk about this a little bit. The experiment was as follows. We take these little particles and we put them onto a little piece of glass, and we stuck them in a chamber, and that's shown in this slide here. And that's a dosing chamber. And this was work done by Frederic Kuhnen and Tom Schmedeke in the lab a couple of months ago. The laser beam is sitting about 20 meters away from this chamber. It's basically a laser pointer. And that laser beam is shooting down and hitting these little particles. And the particles have been tuned to have the right color by making them in the right thickness. Just like that oil on water, you imagine when the wind blows, you can change the color of the oil. We do this by machining into the material a certain color. In this case, these were machined to return a laser beam, red laser beam light, and that's why they look red in this photograph. But on top of that, because they're little porous structures and they'll do this condensation thing, they can condense molecules and their colors will change, and we could probe that remotely with a little laser beam. And that's shown here. This really is kind of remarkable data. We expose these little particles of dust to toluene, ethanol, and acetone, and these are what we call VOCs, or volatile organic compounds. They're pollutants that you might find in the environment. Toluene's added to gasoline, for instance. Ethanol, is something that you put into alcohol. It's drinking alcohol. Acetone is fingernail polish remover. And these materials were tuned so that they had this hydrophobic nature to their surface. And just like those polymers that would suck up some kinds of molecules and others wouldn't, our surfaces are made what we call hydrophobic. And so they are more like these organic compounds than they are like water. And so this top trace shows you how we dosed water in and sucked it back out at this line, dosed it in, sucked it out, dosed it in, sucked it out a couple of times. And there was no response. Our little particles still were red. They didn't have any change in their color. But when these little red particles saw toluene, their colors changed. And we dosed them with toluene a couple of times, 1, 2, 3 times there, and brought the toluene back out of the chamber. And we could see these colors changing using that telescope and that little laser detector. And so I think that's where I'll finish up. I just want to point out that the tour I've tried to take you on is how you can start from some simple concept like a transduction, and I tried to illustrate some of the challenges in taking a signal transduction like a voltmeter that you can hold in your hand and measure conductivity of somebody's body, to how do you make that thing act as a sensor for something that you care about. And we also talked about then doing this thing optically with nanostructured materials and taking advantage of nanotechnology to build up the structures and build up the sensitivity and the specificity for what we want. My research group has been focused on biological and chemical materials for some time. The biomaterials area, we have a lot of collaborations either in the neurobiology group with Yukiko Goda and Sangeeta Bhatias in bioengineering. This is work, most of our biomaterials and biosensor work is being done now in collaboration with these groups. Boyce Collins started a lot of our bio work, and it also was started by the biosensing work in general was started by Keiki Dansel in collaboration with Reza Gadiri at the Scripps Research Institute. We've also talked about more specifically about the gas sensor work that people like Ronnie Chung, Jun Gao, and Ting Gao have worked on. And I showed you some data. Those little smart dust particles were made by Jamie Link, Tom Schmedeke, and Frederic Kuhnen for making these photonic crystal materials. And I'd like to thank you for your attention. Yes, we use that same crystal. And so that was meant to illustrate this point that those three molecules that we were trying to sense are organic molecules, and they're these greasy molecules that if they went into a polymer, they'd swell the polymer. They have that interaction, that like dissolves like interaction. We made our little films to be very, what we call hydrophobic, also very greasy types of surfaces. So they present a surface that looks like oil to the outside world. And so if there's an oily molecule floating around, it finds that surface and sucks into it. And so that's, in general, oily molecules. We refer to most of them as a class as volatile organic compounds. There tend to be very few organic compounds that you want to breathe in. And so this is sort of a general broad class sensor that we're illustrating in that. Yeah, that's a very good question. So the question is, what we've been talking about now, or I didn't really get into the whole issue of sensitivity level. That's another big issue in the sensors area. The gypsy moth is probably the best example. That's a molecule, that's an animal that can detect, they know now, its little antennas can detect a single molecule of a pheromone. And so that's an illustration from nature that it is in fact possible to detect a single molecule. And we have several examples from laboratory scale experiments over in the scientific community where people can detect molecules. And the challenge is to take those single molecule detection methods and bring them into something that can be used and fielded as a sensor. I think there's a big area of interest just in the basic research of detecting and looking at small molecules, but also there's a big interest in trying to then put that into something, or put that into a package or some kind of a device or some kind of a technique that can see a small molecule but also could be low power and in a small package and easily manufactured. The techniques that we've been developing don't really look for single molecules. We tend to look for concentrations that are in the parts per million to parts per billion level. Part per million means if you have 1 gram of your substance, that would be in 100 grams of material. So a part per million detection would be equivalent to taking a sugar cube and trying to find that sugar cube, when it dissolves in a tank car, a train tank car full of water. And to be able to detect a single molecule of that sugar molecule in that tank car is a real challenge. And it's a challenge in both areas of trying to come up with a good transducer, but it's also, as I mentioned at the beginning, that this area of sampling and concentrating your sample so that you can see it. If I've got a sensor standing here, 3,000 to 7,000 spores of anthrax is all it takes to is considered the LD50, the lethal dose that would kill about 50% of the people in this room. If you think about then how many spores are actually hitting, my finger at one time, if this room had an LD50 of anthrax, it'd be a very small number. Might be 0, because if it's in an envelope or on your shirt, maybe it's only a certain region of this room that's being really affected. And so that's a challenge really in terms of what we call sample collecting and sample concentrating. And that's why we've been pushing to this what I call the standoff detection, where you can put your sensor out and have lots of them. so that you don't have to have something like a smoke detector that sits just in the top of the ceiling in one point. We call it a point detector. And eventually the smoke wafts over and the alarm goes off. Yeah, that's a good question. So our little smart dust particles, we're just right now just struggling with this problem. We just developed these materials over this last 4 or 5 months. And we know that we can see the return from a single particle that's the size of a human hair, but we had to be pretty close to see it. And we're working with industrial partners now to try to look at this long-range detection. People that for their bread and butter develop laser range-finding techniques that can focus a beam of light very sharply and look at a long distance and collect a return signal on that. But I don't know the answer to your question right now. How many of these particles would you have to, say, dust into this room if you wanted to use them to be detecting anthrax? That's probably not the real approach you'd want to take, because you don't want to be breathing this dust in either. You'd probably be having it on a surface, and so it would be a smart surface that would turn color, or it would be a smart surface inside a duct that's bringing the air into the building that would be probed maybe in a not fairly far distance. Thank you.